ever wondered how can the DNA actually resolve in a thread? Let's take an example like eye color.
Your DNA has the genetic information that codes for the color of your eye. Let's look at how this happens. The information content of DNA is in the form of specific sequences of nucleotides. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. Proteins are the links between genotype and phenotype. Gene expression, the process by which DNA directs protein synthesis, includes two stages: transcription and translation. There are four stages in protein synthesis: transcription, RNA processing, translation, and post-translation processing.
In transcription, DNA is copied to mRNA, which is used as a template for the instructions to make protein. There are three stages of transcription: initiation of transcription, elongation of the RNA strand, and termination of transcription. Initiation. In the nucleus, RNA polymerase recognizes the recognition sites, causing it to bind to the promoter. The RNA polymerase then separates the DNA into single strands, so the template strand can be read in the 3' to 5' direction. Elongation. Pre-mRNA nucleotides are quickly paired with their complementary bases, which correspond with the template strand of DNA. The pre-mRNA moves in the 5' to 3' direction, while the template strand of DNA moves oppositely from the 3' to 5' direction. Pre-mRNA does not contain thymine. Instead, uracil is used as the complementary base for adenine. Termination. When the RNA polymerase reaches the terminator, it signals the RNA polymerase to stop and release from the DNA. Once separated, the two DNA strands come back together and reform the double helix. The newly formed pre-mRNA molecule is then released. RNA processing. RNA processing is the term collectively used to describe the sequence of events through which the primary transcript from a gene acquires its mature form. Very soon after synthesis by RNA polymerase II begins, transcripts from nuclear protein coding genes acquire a 5' cap structure. During RNA processing, both ends of the primary transcript are usually altered. The 5' end receives a modified nucleotide 5' cap. The 3' end gets a poly A tail. Non-coding regions are called introns and other regions are called exons. RNA splicing removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. After the RNA processing, final mRNA strand is transported to cytoplasm to translation.
Translation. Translation is the biological process by which mRNA is translated into protein. It consists of three phases: initiation of translation, elongation of polypeptide chain, and termination of translation. The ribosome has multiple tRNA binding sites: P site, A site, and E site. P site binds the tRNA attached to the growing peptide chain. A site binds the tRNA carrying the next amino acid. E site binds the tRNA that carries the last amino acid. The initiation stage of translation brings together mRNA, a tRNA with the first amino acid AUG, and the two ribosomal subunits. During the elongation stage, amino acids are added one by one to the preceding amino acid. Termination occurs when a stop codon, either UAG, UAA, or UGA, in the mRNA reaches the A site of the ribosome. Post-translation processing. During and after synthesis, a polypeptide chain spontaneously coils and folds into its three-dimensional shape. Proteins may also require post-translational modifications before doing their job. Some polypeptides are activated by enzymes that cleave them. Other polypeptides come together to form the subunits of a protein. Polypeptide synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the ER. Polypeptides destined for the ER or for secretion are marked by signal peptide. Now, proteins are ready to do their job.